Let's get started. So welcome everybody to this webinar organized by the combined part of the AMR Accelerator, so the European initiative funded by IHI. I think I already see some people that I met last week at the AMR conference in Basel. So it's good to see you again and that you have indeed registered through this webinar. This is probably the first one of a series of webinars where we have a very good list of speakers, leading experts in the antimicrobial development field. The reason why we want to do these webinars is also to give people a chance, of course, to talk about their program, but also that everybody can learn, not necessarily, we call it success stories, depends how we define success, but also to learn from other people that how you go from one milestone to another, and at what point the project might have overcome some challenges or maybe not. So I'm also delighted that we have an agenda with a mixed, how do you say, portfolio of classical small direct acting small molecules, but also some other therapeutic modalities, including phages and also some non-traditional antibiofilm agents. So it's a mix of topics. So I hope that you will enjoy this series. Now we have speakers that are part of the AMR Accelerator, but also out from outside the accelerator. So I'm very pleased that our network has grown. I'm looking forward for the presentations. So, all right. So we will go to immediately to the first speaker, Dr. Huseby from Uppsala University. And Doug will be talking about a novel antibiotic class with potent in vivo activity targeting lipopolysaccharide synthesis in gram-negative bacteria. Doug is part of the University of Uppsala and also is within the Enable2 consortium is their project manager and researcher. So Doug, the floor is yours, and very much welcome to hear about your story.
Right. Thank you for the introduction, Frederick, and the invitation to speak. I'm going to be presenting our project developing LpxH inhibitors. And just to give you a little context, the sort of angle that this project is going to be coming from is that this is a project that was developed in an academic context. But in spite of being developed in an academic context, we were really quite focused on the idea that we want this to eventually be out in the world. So that was the focus, is that we're not just trying to develop an antibiotic that's going to work in the lab, but rather that we're going to be making something that could eventually be out in the world and really focus our development in that direction. So I will just say that the team that we had working with us was a academic team with multiple different specialties working on this project together, which is a little unusual, and I'll talk a little bit later about how we were able to put something like this together in an academic context. But this is a little unusual because typically in academia you'd have one group working on it, and so then you would bring that group's expertise, but wouldn't necessarily have all of the other expertise that you might need to develop antibiotics. And we were really focused on developing something with gram-negative activity. So we were initially screening compounds for gram-negative activity. So I could say we started by screening various sources for things that might have some gram-negative activity. And in this, I'll give you an example here. We were looking at a group of things that had activity against efflux defective gram-negative bacteria. And we ended up with about 1,400 compounds that were active. And from there, we were able to find 273 of these that were commercially available, and we got 74 of those, and eventually we winnowed it down to this compound Jedi 852, And we resynthesized and retested this compound, and were able to synthesize or select resistant mutants in and determine that the target of activity of these compounds was LpxH, and we could map the product to the substrate binding site of LpxH. So LpxH is an enzyme that's in the lipid A synthesis pathway, and lipid A it goes to form the LPS, which it forms the outer surface of gram-negative bacteria. So here in the lower corner here, you have a schematic of a gram-negative organism, and zoomed in, you can see the LPS here on the outer surface, and lipid A is synthesized in the cytoplasm of the cell, then it's eventually transported to the outer surface of the cell. So this compound, JEDI 852, was identified from a PubChem screen of compounds with activity against efflux defective E. coli. But importantly, it didn't have any activity against anything other than efflux defective bacteria. So no wild type activity. And this is something that is critical and could be something that could be difficult to overcome. So this was going to be a key liability that we were going to need to address. From looking at compounds that had activity against LpxH, we identified a compound that had previously been determined to have activity against the enzyme LpxH, which was this compound called AZ1. But it had a similar liability, that it didn't have any activity against wild type bacteria. It only had activity against efflux defective bacteria. So we went to making analogs based on JEDI 852, but we didn't produce any breakthrough activity against wild type bacteria. And so this was going to be something that we were going to need to work on if this was going to be developed further. But we did notice when looking at these two compounds that there was a shared structural motif between AZ1 and JEDI 852. And we were able to combine the shared structural motif here. We have it highlighted kind of in purple. It may be a little bit faint to see, but perhaps you can see it. We were able to combine these compounds into this compound JEDI 1444. And amazingly, we picked up wild type bacterial activity. And so this was a really important development for us, because this meant that we could actually develop this towards being a real antibiotic. So I'll tell you that JEDI 1444 was quite a breakthrough for us, that it had wild type MIC activity, but it did have a number of liabilities that we were going to have to address in order to eventually get this further. So I think a lot of these are quite interconnected. So we have the C log D. It had a very high C log D, and it had poor metabolic stability here, as exemplified by the mouse liver microsome and human liver microsome stability. It had poor solubility. So we were going to have to solve that. But again, that was probably connected to the C log D. And so all of these things were going to have to be addressed in order to continue to develop this. So I'd just like to say that we also had, as a part of our development, we had access to crystal structures, which turned out to be really, really important for the development of these compounds. So in the lower left corner, what you can see is that you have a schematic of our compound complex to, a schematic of the product of the LpxH reaction, lipid X, in LpxH crystal structure that we and our collaborators put together. So you can see that you have this compound lipid X, which has two long aliphatic chains, and one is buried inside of the enzyme, and one is sort of exposed to the solvent on the outside of the enzyme. And up here, where you have these manganese ions, this is sort of where the catalysis actually happens in the real LpxH enzyme. But then over here on the right, what I'd like to direct you to is we still have the same lipid X, and now we've cut away a lot of the protein structure except for some of the key interacting amino acids. And in blue here, now you have our compound Jedi 1444 overlaid with the LpxH. And what you can see is that buried aliphatic chain, this orange chain, forms kind of a 90-degree turn inside of the enzyme. And what's very cool is that we can see that our compound, this Jedi 1444, mimics this turn and actually has some interactions with the amino acids around there. Some of them are similar to what the actual product interactions, and some of them are quite different. But because of this crystal structures that we had access to, we were able to really do structure-based drug design throughout the process and really tailor our compounds to try and make sure that they were going to have the best chance of success. So, I've told you that Jedi 1444 was far from a useful drug based on the poor drug-like properties of the molecule. We went to work optimizing the molecule to improve these properties. So here's where we're at. We have wild-type bacterial activity, but we lose activity in the presence of serum. We're poorly soluble, and we're metabolically unstable. So through a lot of optimization, this really compresses down a great deal of the work we did in making many analogs to test different hypotheses and see where modifications could be made on these compounds. We found that there were some options to make modifications to ring 1, and so we screened some ring 1 variants of these, and we found that we could come up with variants in ring 1 that improved the solubility, but they retained the liability of poor metabolic stability. And then over here on the right, we found that by building out on this ring 4, we could add some extensions, cationic extensions, and these improved the metabolic stability and improved the activity in serum, but still had the liability of poor solubility. So I'm sure many of you can probably guess what we were able to do with this, which was by combining these modifications on ring 1 and cationic extensions on ring 4, we ended up with compounds that actually sort of had much better properties than any of the individual compounds. That we had potent antibacterial activity, and we retained activity in serum, and we improved a lot of the other physiochemical properties of these compounds. So these are two compounds that we went forward with, EBL 3647 and 3599. So just to give you sort of, Jedi 1444 had a number of poor properties highlighted here in red. And I think you can see by just the colors here in 3647 that we were able to fix a number of these properties, or at least improve them to a point that we were happy with. You can see that the C gone down quite a large amount. We have rather good now, and the C log D was corresponded to improved solubility, and importantly, we now had activity in the presence of serum, which suggested that we might have something that would be useful in vivo. So that's exactly what we did. We took these compounds and we tested them in an in vivo murine peritonitis model against E. coli and Klebsiella. And here I'm showing you the results for that experiment for E. coli. So at the top we have our CFUs after the experiment in peritoneal fluid, and on the bottom we have in blood. Not sure what you heard. Hopefully you were able to see the slide. So maybe you've gotten the point anyway. On increasing in the case of the blood CFUs here, we were able to go below the limit of detection for the higher doses. So it's really promising from the point of view that this validates that compounds targeting LpxH can be viable in vivo, that they really work as antibiotics, have some promise in the future as antibiotics. So I'm a microbiologist, so I just like to bring up this data, which is interesting. That one of the assays that we would do is to look at McK90s. So this is an assay to see the susceptibility of a large set of clinical isolates. And what we would expect for any antibiotic that has been out for a while and has seen some resistance development is that you'd see maybe a bimodal peak, that you'd see some liability towards the high end. that you'd have some isolates that would be non-susceptible at the highest end of concentrations. But what you can see for ours, which is expected for a compound that has never been used as an antibiotic before, is that we have a unimonal distribution, a fairly narrow distribution of mix, indicating that these compounds, at least there's no pre-existing resistance to these compounds. So I said that our group, our consortium was a academic, and so but importantly, we've done this development compounds should hopefully eventually be able to be real antibiotics. So we've done this in a way that we're protecting our ability to continue developing these antibiotics, while also trying to share what we've done with the community of people developing antibiotics. So trying to balance these two things really. So I'd just like to conclude by saying, the compound Jedi 1444 was the first LpxH that we type bacteria, type E. We optimize this compound to improve the physiochemical and ADME properties, outside of the in vitro microbiological properties, which led it to have sufficient properties that we could then test it in vivo. And I'd just like to add that development chemical series is ongoing in the Enable-2 gram, and we're trying to optimize the activity, spectrum of activity, the route of administration, and of course, a big issue with a lot of antibiotics to minimize potential toxicity. So I'd like to acknowledge that a lot of the work that was done here was done in the New Drugs for Bad Bugs Enable program. So this was an IMI program that existed several years ago that helped a lot of antibiotics develop. Following the end of Enable, Enable-2 began, which is kind of a follow-on program that is funded by the Swedish Research Council in an effort to sort of continue the momentum that Enable had. And so the idea is that this is to marshal academic expertise in developing in laboratory techniques and ask advice from people who have developed antibiotics before to try to accelerate projects to get them to a place where maybe they can be picked up for one of the later stage initiatives to develop antibiotics. So the Enable-2 project and Enable in the New Drugs for Bad Bug Enable had a lot of collaborators. I put some of them down at the bottom here. So this work has been helped a lot by contributions from SciLife Lab, the Latvian Institute of Organic Synthesis, LIOS, SSI, and RISE. So I'd like to just take this opportunity to let you all know about Enable-2. So Enable-2 is the initiative that we're working on, and it supports early stage antibacterial drug discovery projects with cost-free experimental support and advice. And so if you're interested, if you're an academic group within Europe, you can contact us, and we're happy to have conversations with you about whether your project is eligible and what we might be able to do to help you. So thank you very much.
All right, so our next speaker, let's move on. Thanks again, Doug. And I would say let's applaud, but it's a virtual meeting, so congratulations with your presentation. So the next speaker, if we can have your camera on, then we can do a quick check, is Lorenzo Corsini. Hello, welcome. So Lorenzo, your talk will be different, right? So as we said in the beginning, it's a different talk, a different success story, also different therapeutic modality as you are developing antibacterial endolysins. And we're looking forward to hear your PhagoMed journey, as you call it. So welcome to this session.
Thanks very much for inviting me. First of all, I'm very excited to give this talk here. It's gonna be very different in that I'm gonna focus much less on the science and much more on how we actually build PhagoMed as a company and what sort of choices we were facing, how we thought about those choices, how we prioritized, and how we went on. So yeah, PhagoMed. PhagoMed is the German pronunciation. I guess when we came up with the name, we didn't think about that probably English-speaking people wouldn't really know how to pronounce it. So it wasn't that's maybe the first lesson. Don't choose a name like this. But in the end, yeah, PhagoMed doesn't exist anymore. It's now BioNTech Austria. But it's the company that I founded together with a number of co-founders. It was a super interesting journey. I learned so much. And yeah, I hope it's going to be interesting for prospective founders, and hopefully then anybody in the AMR space. So the story begins in 2015 when I was a consultant at the Boston Consulting Group. I'm a biochemist with a PhD in molecular biology, and I always wanted to start a biotech business. And there were many ideas and some apps and softwares, but it all seemed a little bit incremental, and I couldn't, let's say, show enough passion for anything to really quit my job and start it. Until that is, I met Alexander and Burkhard Wippermann, my co-founders of PhagoMed. Especially Professor Burkhard Wippermann is a orthopedic surgeon. He at the time was doing probably 400 surgical interventions on hips and knees and all types of prostheses per year. And he really introduced us to the space, the AMR problem. But then more than that, really, from an orthopedic surgeon perspective, there is this problem of infected prostheses, of biofilms that cannot be treated by any antibiotic, irrespective of antimicrobial resistance. So the appropriate treatment is surgical removal of the biofilm. There is no known drug that can cure a patient from a biofilm. And in last resort cases, he had experience with using bacteriophages sourced somehow from Georgia in a not standardized way in non-GMP. And he had the impression there is something behind it, but really he wanted to help more patients with a more standardized drug, with an actual drug produced in GMP and regulated under the appropriate regulations to have a standardized therapy. And phages somehow seem to be well a good bet to develop a drug because it's the most abundant biological entity on Earth, very specific, so also known to be quite safe. And so we really set out to try to find a match between the high medical need and phages as a technology where we didn't really know exactly what to use it for at the beginning. So that's us three founders. As I said, we started with a vision to help patients by developing safe and effective drugs against bacterial infections. However, we also felt really strongly that we would have to that helping patients means developing products that are financially sustainable, and that would mean improving over the standard of care, not just being non-inferior to the standard of care, which would mean superiority trials, and that meant really thinking very hard which indications to bet on, and so we systematically looked at what are such indications where it's conceivable to improve the standard of care. Where is it conceivable then to do superiority trials? Where is it conceivable to do trials at all with realistic recruiting and enrollment numbers? Where is the medical need high enough in terms of number of patients, but also the severity of the disease itself? where does it even microbiologically make sense to apply phages? Do you have a clear causality between the indication and the bug that you can target? Is the route of administration feasible? All that. In the end, we landed on these three that you see on the slide: prosthetic joint infections with Staphylococcus aureus as target, complicated UTI with E. coli as a target, and bacterial vaginosis with Gardnerella vaginalis as a target. And we focused on the first two, with bacterial vaginosis being a bit of a back burner, with least capital allocated to it. And so mainly to start with a portfolio approach, because phages are so new that we couldn't really bet on, or we thought we shouldn't put all eggs in one basket, and rather, at least initially, cover a little portfolio of indications rather than just one. So we wrote target product profiles for each of those and defined in very high detail and set ourselves quantitative targets for what we wanted to achieve in the different dimensions, from activity over PK over stability over whatever, for these three indications with experimentally verifiable specifications for all three. Then we started to work, and initially it looked really good. So we got funded. We raised 2.5 million in private investments. We got quite substantial grant money from the Austrian government mainly. We had a great lab and office actually as subtenants of another antibiotics development company, Nabriva, that some of you may know from the development of lefamulin. We had a great, motivated, very talented team working on this. We were starting to write papers and patents. So we did some targeted, directed evolution to improve over natural phages, and had then mosaic phages that actually could be patented. We were getting some traction in terms of publicity. We won some prizes, and Alexander gave a TED talk, and there were some news outfits reporting on us. So it was looking really good. So, except that probably 2019 already, there were some clouds at the horizon, in the form that most experiments started to show that the phages just wouldn't meet the specifications that we set ourselves in the target product profiles. And what I mean is exemplified on this slide. Actually, it's published, so if you want to read up all the scientific details, you can do this. One problem was that the phages we were testing just were not active against or were not active in the presence of plasma or synovial fluid. And what you see here, this chart, all the subcharts are basically time series of bacterial growth, with the horizontal axis being 48 hours, the vertical axis being optical density between 0 and 1. And you see here in the bottom row, the normal growth, the undisturbed growth of bacteria. And in the rightmost column, you see what happens if you add the phages. So bacteria don't grow anymore; they're dead. That's what the phages are supposed to do. And over the course of 48 hours, we also didn't see any resistance formation, what you see with some phages. So we actually had good phages. Problem: by adding even minute amounts of plasma or synovial fluid or interstitial fluid or anything really that contains elements of the coagulation system, the phages were basically dead. We analyzed it deeper. So basically the absorption of phages to Staph aureus and to some other staphylococci is strongly impaired, is reduced by orders of magnitude in the presence of plasma, which means that the rate of bacterial killing by the phages just drops much below the rate of replication of the bacteria, essentially making the phages useless. And this is what we saw with our other, we had other technical problems with the other phage programs as well, and so we were starting to become really worried. On the other hand, we had one endolysin program against Gardnerella because at the very beginning we were just not able to find phages against Gardnerella, so we could just continue with an endolysin clone from a prophage. Then we did a directed evolution screen. We're able to file a patent. So we had a good endolysin against Gardnerella, which was sort of the backup of the backup program. But there, the protein was meeting the specifications of the TPP. What you see here, again, it's published, so you can read the details of this in actually multiple scientific publications. Just here, some data to show you the core of it. The lysine, which we now call BNT331, is highly active against bacteria of the species Gardnerella vaginalis, Gardnerella leopoldi, Gardnerella sudzinski, actually all species that we tested within the genus Gardnerella, while it was not active at all against beneficial lactobacilli, which constitute the beneficial microbiome of the human vagina. The MIC range of BNT331 is actually very low. Didn't find any resistant strains. The range is very comparable, also the MIC90, very comparable to what we measured for clindamycin. Clindamycin and metronidazole being the two antibiotics used against bacterial vaginosis. But here already you see metronidazole has a resistance problem. So MIC90 is in the resistant range because there are so many strains that are already resistant. So bottom line, we were facing a fork in the road. The main highway being the phages, that's what we built the company on. That's what we named it after, PhagoMed, right? That's what we raised money for. That's what we got the grants for. That's what we were always pitching. But we really felt, if we continue full speed on this road, we might actually hit a brick wall. On the other hand, there was this fork off that was looking much less clear because we had only one lysine. We didn't really want to become a single asset company. We wanted to stay a platform company, but it was unclear whether we could repeat the early success of the Gardnerella lysine also with other lysines. So we had discussions with our investors, amongst ourselves. It was really tough. Should we continue on both roads? Should we invest just a little bit more on the phages and then we're going to solve the problem? Ultimately, we decided to drop the phages completely. So from 1 week to the next, we shifted the whole team on license to show that we can find more license against other indications, against other bacterial infections that would fit the criteria that I mentioned at the beginning, that would be suitable for a lysine development for superiority trials. And that was actually quite successful. So that it worked relatively quickly, that we had a whole portfolio again of endolysins, which led then in 2021 to, initially we wanted to raise money in a classical Series A, and that somehow escalated into full acquisition by BioNTech, and so now we're not called PhagoMed anymore. We renamed the company into BioNTech Austria, and the name PhagoMed disappeared, but it's still the same company. We're still an independent entity within BioNTech as a larger house. And really, at the time, we felt the vision still being helping patients with bacterial infections that cannot be treated sufficiently with antibiotics. And we really felt the probability of technical and regulatory success as a part of BioNTech would increase compared to what we could do as an independent small business. Especially because 2021 was after the the huge success of the COVID vaccine. So using some of the funds generated with the COVID vaccine to work on antimicrobial resistance and biofilms, that seemed to be a really good idea. It still is a good idea. Also, just leveraging the huge expertise and experience of BioNTech in terms of clinical development. That's something we just couldn't have done had we gone alone on it. Another aspect of the whole story was that we were planning to develop endolysins as ribolysins, and unfortunately I can't go into more details than what's written on this slide. You'll find the same information in some press releases and a patent application. But bottom line, that endolysins are proteins with a enzymatically active domain and one or more cell binding domains, and for gram-negatives, sometimes you have to add these outer membrane penetrating peptides. But being a protein of size 20-ish, 15 to 30 kilodalton, the pharmacokinetics is really bad. Right? The half-life in plasma is something like between 20 minutes and an hour. The renal clearance is very high. And bringing these proteins on mRNA and injecting the mRNA instead of the protein has certain advantages in terms of bioavailability and pharmacokinetics. And in antimicrobials, PK/PD is really everything. So improving the PK along with the actual activity of the protein is something extremely important. Then there are some other advantages of mRNA formulations, such as the formulation that's on one platform. So you could envisage having more than one endolysin that's all formulated the same way, that's all manufactured the same way with the same CMC process, same basically GMP process. And so that's something that that we're working on. Fast forward in 2024, the program that actually started, as I said, as a backup of a backup, the Gardnerella endolysin that by now is called BNT331, is now being tested in a clinical trial. It's a combined phase 1a/b trial that tests safety, obviously, but also pharmacokinetics and pharmacodynamics, and also cure, basically, of BNT331. In part A, that started recruiting last year, that was designed as a single ascending dose in healthy women, 24, with a 3-to-1 randomization, BNT331 against placebo, 3 doses or 3 dose levels. So from this, you also see that we solved the GMP issue, and we solved the issue of how to formulate a protein from E. coli or to formulate the protein produced in E. coli in a solid vaginal insert in GMP. So part A finished recruiting end of last year. So we proceeded with part B, which is a multiple ascending dose study in women with bacterial vaginosis, recruiting 72 women with a 2-to-1 randomization, again BNT331 against placebo, with a 5-day treatment, 5 daily doses, and 2 different dose levels. And this study is gonna finish recruiting soon, and we're gonna see data this year. So a couple of thoughts on this whole journey, and what at least what I learned from it is that phages are still scientifically extremely interesting, but in the whole time since we started working on this back in 2017, the therapeutic value still needs to be shown. There are so many case studies. Safety seems to be fine as long as the phages are produced properly, but efficacy is really the question. and efficacy was also the reason that we stopped it. If you read the literature on what are potential barriers for phages, that typically includes that phages can be produced in GMP. That's just not true. Phages can be produced in GMP. Many companies showed that, and studies were conducted both under the jurisdiction of EMA and the FDA. So for both authorities, producing phages in GMP was apparently possible, and they were apparently happy enough with the data provided that they allowed these studies to proceed. Often said regulations hamper it. There is no clear path to a marketing authorization. So yes, it's a bit unclear, but all the authorities we talked to were very supportive, both in Europe and in the States. They encouraged us to produce data, and then we will find a way. It's always said that phages can't be patented. It's also not really, because we developed mosaic phages that were completely new, improved over the wild types, and that we filed patents for. We also got funding. So really the problem was we couldn't make the phages meet the specifications of our TPP. And then the choice was, okay, either relax the specifications, but then we doubted that this could be a viable product, a technically viable product, also a financially viable product. Or actually trust the data and stop it. And so in contrast, our experience with endolysins is very positive, both from a technological and a regulatory standpoint. That seems to work, seems to be easier. Another aspect that I feel even more strongly about than when we started PhagoMed is that in AMR you need a path to superiority, at least a vision, at least it needs to be conceivable to improve over the standard of care, because you will need financially oriented investors, and pull incentives are just still way too far away to take the private financial risk to fund something like this. And unfortunately, we're seeing so many technically and scientifically highly successful companies fail because the path is so narrow. And we always try to not even use the word antibiotic or antimicrobial resistance. Our vision, not saying we can do it, right? That clinical trials will need to prove whether we can do it, but the vision was always superiority. And
we conclude here, Lorenzo.
Sorry?
We'll need to conclude here.
This is my last slide. So my last learning is just trust your data, work on TPPs, use the TPPs to also kill a program if it does meet the specifications. And in our case, that really saved our company. Thanks very much.
Thanks for this really very interesting, inspiring talk. Thank you very much for this.
Thank you.
Okay, so our third speaker for today is, again, as I mentioned in the beginning, will be, again, something different. Michael, I will not give you a very long introduction, but what I'm intrigued, and I think we talked about it at the AMR conference as well, is that I think you will tell it, one of the first, with your team, of course, that we're looking into non-traditional and antibiofilm agents. And it has been also, I'm sure, an interesting journey. So we are happy to hear your story. For those who do not know Michael Graz, so he's CEO of Disperazol Pharma in Denmark, and also CSO of Biofis Limited in the UK, as a research fellow at Costerdon Biofilm Center at the University of Copenhagen. The floor is yours.
Frederick, thank you so much for the invitation to speak. I suppose being the last speaker has its advantages, because both Douglas and Lorenzo have said at least half of what I was going to say, so I'll make it short and sweet. But realistically, just like Doug's introduction, my project started out in academia from a consortium that included the University of Copenhagen, DTU, and at times the Salsi Institute in Singapore, before the technology I'm going to speak about was spun out. And I also have to give a punt to Enable too, because during the development of Disperazol, which will be the second project I'm going to speak about, we have had the opportunity to engage with Enable. And even though our approach is not a direct acting molecule, they have given us very sage advice in terms of our medchem. And just like Lorenzo said when he started out his discussion, I have the same view that there are no drugs that target biofilms. And herewith starts my story. My story starts 15 years ago at the Costerton Biofilm Center at the University of Copenhagen. And it really started out as a fundamental question on how are Pseudomonas biofilms controlled, and can you disrupt that control? And that control was looked at from two different perspectives. The first was looking at quorum sensing inhibition, or specifically quorum sensing to start with, which is, for those of you that aren't familiar with it, an extracellular, intercellular signaling or form of signaling was probably first described or extensively described by Bonnie Basler, I'm going to say 30 plus years ago. And then the second approach is looking at intracellular signaling, and very particular to gram negatives, we were looking at the intracellular signaling molecule cyclic di-GMP and how its modulation within a cell could possibly be controlled, and how it would then affect biofilm formation. And we had this opportunity, which sort of fell into our lap, or should I say more specifically into our basement, that we inherited a 60,000 compound, or should I say natural compound library from Novo, which had originally been created to look at GLP-1 activity. Well, we chose to use it for something else. So it all started with this quorum sensing inhibitor, and just very quickly, an increase in quorum sensing leads to a more developed biofilm, which provides this biofilm and the bacteria within it with a very strong mechanical protection against environmental insults, and increases the virulence of the bacteria. And by decreasing quorum sensing or inhibiting the pathways in some way, you reverse all of this. And this is where NXAS401 was born. And right, why am I not moving? There we go. So the molecule was spun out of the University of Copenhagen probably just under 15 years ago, and it specifically targeted the quorum sensing signaling pathway in Pseudomonas. We had a validated mechanism of action. It blocks the production of eDNA, which the bacteria use to prevent external insults by binding specific antibiotics, and quite frankly, that in itself is probably subject to a separate lecture, which I won't go into. But at the same time, it also affects the production of the diramnolipid that Pseudomonas produces, and that rhamnolipid quite particularly disrupts and destroys PMNs, and therefore very much inhibits the efficacy of the immune system. So if you are able to prevent that, you're actually facilitating an immune response. But most importantly, we had no direct lethal antimicrobial activity, neither in Pseudomonas nor, for that matter, in Staphylococcus at the proposed therapeutic doses. And we were able to improve the efficacy of our molecule with very targeted rational design, which was done in Singapore on our behalf, and I'll get back to that in a moment. And again, this is not quite doing a thing. So without spending too much time on the data, because I rather want to speak about our journey, confocal images of the treatment of Pseudomonas biofilms in a drip flow reactor showed the significant effect of a combination of our molecule together with tobramycin, which will become evident in a moment, in both the laboratory strain, which I suppose we're all very familiar with, which is PA01, and clinical isolates, which we had inherited from the University Hospital in Copenhagen. And the important thing for us is that a non-lethal agent will always be used as a co-therapy. And so all of the work we've done has always been together with the standard of care antibiotic of the indication that we were targeting. But more particularly, looking at an in vivo model, and very particular, we looked at an intranasal installation of Pseudomonas into a mouse model. And by this stage, you can probably guess we were targeting lung infections. But what we were able to show that with a single dose of our quorum sensing inhibitor together with tobramycin, we were able to show a trend of decrease in the nasopharyngeal reservoir of Pseudomonas, which where the combination was significantly more effective than tobramycin on its own after a certain period, but as you moved on, tobramycin then became as effective on its own. We used multiple models to test this, not only a nasopharyngeal model, but agar beads implanted into the trachea, thigh infection models, and implanted foreign body models, and in all cases we found similar results. And that led us to believe that we could do something with this and actually start developing it as a drug. And as many of you will very well know, that as an SME, you can't do it on your own. So we started putting together this ecosystem of both partners to do our development, but at the same time also of funders. And the project was originally funded by the German Cystic Fibrosis Association, or the Mukoviscidose Institute, which brought together a whole host of people that could help us in our development. And as I said, we were funded by the Mukoviscidose Institute. When the molecule was then spun out, the translational work was funded by a family office. And I think in a lot of cases, this is what happens. In this case, it was a family office with a lived experience in cystic fibrosis, and they wanted to see a potential new antimicrobial against the lung infection that had caused the bereavement of a family member. And so we targeted Pseudomonas in lung infections in people with cystic fibrosis, and at the same time, knowing full well that a single indication would not be sufficient, we also considered ulcerating wounds which were positive for Pseudomonas. And in 2015, with only one corrector on the market, for cystic fibrosis, targeting lung infections in CF, had a very significantly positive NPV, as had been worked out by Cogentia for us, and was very attractive to our investors. If you were to ask me that question now, with the plethora of correctors that are on the market, I would say that such a narrow indication is no longer on its own a viable proposition. But really, when developing a non-traditional, I think what you really need to consider is, what is the regulatory pathway that you're going to follow? And 10 years ago, neither we nor the regulators really had an idea. But I'll share some of our learnings with you. And it's probably a litany of do's and don'ts more than anything else. And the first thing I can say is, get expert advice before you meet a regulator. Talk to someone who's been there or who knows. And there are some fantastic organizations out there that can help you. And I wish we had known that 10 years ago. Because when you meet a regulator, you need to know what questions you're going to ask. because they will only answer the questions you've asked them, and generally in writing before the meeting. Then also as an SME, make sure that you have SME status if you qualify for it, because it has a very significant effect on the costs that you have to pay. Otherwise, it becomes exorbitantly expensive. If the regulators, and be that the FDA or EMA, have issued a guidance with respect to the type of indication that you're pursuing, make sure that you've read it and that you followed it. Otherwise, they will point you back to repeating the work that you've done to follow the guidance, which may be significant cost and a significant time loss to you. The other thing that we learned was make sure that you can explain your mechanism of action in what I'm going to call layman's terminology. Don't expect that the people that are across the table from you understand what you're doing. They're not on the same plane as you are when it comes to the mechanism of action of your molecule. And we had to learn that the hard way too. And I suppose for us, the most surprising thing was expect contradictions in the same meeting. We had a meeting with the MHRA in the UK, and we're talking pre-Brexit, when they were still very much the rapporteurs for EMA. And literally two experts in the same meeting had an argument about the efficacy and the mechanism of action of our molecule in front of us, which was highly unexpected, and there was nothing we could do except sit there and listen to this and not become any the wiser. Also, don't expect a definitive answer at the end of a meeting. Expect answers and guidance. The only time you're going to get a definitive answer is whether or not you get a marketing authorization or not. But last but not least, I'm going to say, these meetings do sometimes need a bit of luck. When we went to the FDA with our quorum sensing inhibitor, we were very fortunate that the person who assessed our program came from Montana State University, which is the home of the Center for Biofilm Engineering, where quorum sensing was originally identified. He understood what we did. And so the benefit of that was we were the first organization to get orphan designation for a quorum sensing inhibitor against lung infections in persons with CF by Pseudomonas. It was an amazing success, which we celebrated very extensively in the press. And unfortunately then this happened. Our program died. And unfortunately what we should have done is very seriously considered the toxicology of our program at therapeutic doses really early. When treating our mice and the cell cultures that we did our infection models in, we never detected any significant toxicology. However, our molecule had a disulfide bridge in it, and for those of you that can guess what I'm going to say is sulfur and DNA do not good bedfellows make. It was highly mutagenic. And the program was stopped literally while we were in IND-enabling toxicology. That unfortunately obviously was really bad news for us. It was very sad for our investors who had put a lot of time, effort, and a lot of money into the program. And unfortunately, it also led to the end of the business that was developing this molecule. But in all cases, it was a significant step forward because quorum sensing inhibitors were recognized as a potential modality for treating lung infections. And so we pivoted. Disappointed? Yes, completely. Discouraged? Not at all. And so we started looking at cyclic di-GMP, which in high concentrations within the cell leads to the formation of biofilms, and low concentrations of the cell leads to the dismantling of biofilms or the prevention of biofilm formation. We learned from our previous experience, and what you may notice is that this slide is significantly more populated. We found ourselves an enhanced ecosystem with more people to guide our steps. We ensured that our funding streams were solid and not dependent on a single organization. And just like Lorenzo, a lot of our work, we've been very fortunate to be funded by grants with small investments into our company. So what is Disperazol, as we call it, which is a molecule that came out of that screen of the 60,000 compound library from Novo? Well, it targets cyclic di-GMP. It's a pyrazole scaffold, which after very significant SAR, we got to what it is today. And the molecule, which I can't see my whole screen, is not on the screen. I do apologize. But it is well published and very happy to share it with you. We know that it depletes cyclic di-GMP. It causes the dismantling of pre-established biofilms, and it is very, very particular in terms of being dose dependent in that activity. It again has no direct acting antimicrobial activity, and something we obviously did really early on, we sent it to an independent third party for toxicity, and it came back to suggest that there was minimal toxicity at the proposed therapeutic doses, which led us to continue with the development. We know what the direct mechanism of action is, but thanks to the NL lab at the University of Basel, our molecule actually affects a phosphodiesterase called BifA. And the identification of this has now led us to do a fair amount of rational design for cyclic di-GMP inhibiting compounds to target other gram-negative bacteria. And seeing that we started with CF, we looked at CF again. And should we have the opportunity, we could possibly target lung infections again. Just very quickly, if I can get my screen to change, there we go. Very similar data. If you look at the left-hand side, left-hand side of the screen, in block A, we can prevent the formation of a biofilm over a 48-hour period. If you look in the center of the screen, we can dismantle a preformed biofilm within 4 hours, just in the presence of Disperazol with no other antimicrobial agents. And on the right-hand side, probably my favorite picture, that if you stop the stack for a moment and you take a photograph, you can actually see the bacteria which are being lifted from the surface of a biofilm. And then fortunately for us, I'll skip that. We have collaborators at the Biozentrum from the NCCR anti-resist program in Basel. And a week ago in Basel, Dr. Flores from the Dehio group actually presented some work using Disperazol to remove Pseudomonas biofilms, both lab strains and clinical isolates, from from human bladder organoids using their system, again, only using Disperazol. And on the left-hand side, you can see the bacteria. They're sort of highlighted in silver using the way that the computer has highlighted the size of the aggregates. And then if you look at exactly the same image again in the presence of Disperazol, those aggregates are removed. And again, the same thing happens with PA175, which is a clinical isolate from the University Hospital in Basel. The great thing for us is this showed to us and demonstrated that Disperazol works in the presence of human tissue, but more importantly for us, a completely independent third party showed the exact same results that we had found. So in this case, we thought, well, now what do we do? How do we select an indication? We have an active molecule. We know that it reduces. Yeah, that's fine.
2 minutes.
I'll be done in a second.
All right.
How do we select an indication? Well, we had an active molecule. However, we were targeting catheter-associated UTIs, which is a very, very small group of patients. And for those of you that were in Basel, a week ago, very eloquently put by the CMO of Bioversus. UTIs are maybe not the best place for an SME to go unless you have very deep pockets. When you get to marketing authorization, you are still competing with generics, and the chances are you are not going to make your money back. And very last but not least, I will get back to the slide. If it hadn't been for the advice that were given about a year ago, we would be at that point about to go into preclinical, or should I say into IND-enabling toxicology for first in man. However, we have decided, because of the narrowness of our indication, that we are going to try and expand the efficacy of Disperazol. So we've gone right back into the discovery program to see that we can expand the spectrum of bacteria that we can treat and look at other indications beyond the very narrow Pseudomonas catheter-associated UTIs. My conclusion, I think that non-traditional, non-lethal agents do have a future. More engagement with regulators is very much necessary for them to understand what it is that we do. And I thanks very much our thanks to the Beam Alliance for engaging with EMA specifically on this topic and starting to get some regulatory advice and some scientific advice of what would be expected. I think very, very much, both from Lorenzo and my experience, choose your indications wisely. And I think the last but not least is, don't stop innovating, because I think that's where we're going to find the new agents, and whether they be non-lethals like ours, or hopefully the other approaches that were mentioned before me. I think that is the opportunity that is presented to all of us, and it's the biggest challenge that we all have. And with that, I thank you.
Thank you so much, Michael, again for a wonderful presentation. As we mentioned, these are three completely different presentations and products, at the same time, very challenging ones. And so, Doug, Lorenzo, Michael, thank you very much, and we'll see each other again, as this is a very small community, as we saw each other last week as well. So, but thank you very much for your talks, for your presentations.
Thanks for inviting me. Thanks for
Thanks
attending
again
our
for
talk.
inviting us.
Thank you.
Bye.
All right. Bye.